Hello and welcome everybody to my channel MSc Entrance, and today we will be discussing GAT B question paper conducted in the year 2022. If you are new to my channel, please do like, share and subscribe. Coming to the first question. These are questions from section A and I will be discussing only the biology questions. So coming to the first question. Given below are two statements. One is labeled assertion A and the other is labeled as reason R. In the light of the above statement, choose the most appropriate answer from the options given below. So assertion A. Hematopoietic stem cells are considered multipotent precursors. Reason, they have the ability to differentiate into different type of blood cells. So both these statements are true and R is the reason for A. So the answer here is option 1. Both A and R are correct and R is the correct explanation of A. Then glycosidic linkages are bonds between. So glycosidic linkages are bonds between sugars. Option 1. What is the bond that can form between two cysteines in a protein? So between two cysteine residues, it is always a disulfide bond. So the answer here is option 3. Cystic fibrosis is caused due to. So cystic fibrosis, it is an autosomal recessive disease and it is caused by a mutation in the gene cystic fibrosis transmembrane conductance regulator. And this particular gene, cystic fibrosis transmembrane conductance regulator, it belongs to a large superfamily of ATP binding cassette transporters or ABC transporters. So the answer here is mutation in the gene encoding an ABC transporter. Answer is option 3. Then next one. The primary enzyme responsible for the rapid and accurate replication of the genome in E. coli is. So E. coli has got 3 DNA polymerases, DNA polymerase 1, 2 and 3. Of these, the main replicating enzyme is DNA polymerase 3. So the answer here is option 3. Histones are tightly bound to DNA because histones are positively charged proteins and are usually rich in positive charged amino acid, that is lysine and arginine, and DNA is negatively charged. So that is the reason why histones are tightly bound to DNA, because they are positively charged proteins. Answer is option 1. Then next one. Given below are two statements. In the light of the above statements, choose the most appropriate answer from the options given below. So the first statement, a single gene can affect multiple phenotypic expressions, and in a polygenic trait, multiple genes contribute to a phenotypic expression. So a single gene can affect multiple phenotypic expression, that is correct. And a polygenic trait, multiple genes contribute to a phenotypic expression, that is also correct. So both the statements 1 and 2 are correct and your answer is option 1. Interferons are secreted by virus infected cells induce. So interferons secreted by virus in infected cells, they induce antiviral response. Answer is option 1. Lung cancer can be detected at an early stage by. So it can be detected by MRI scans. Answer is option 3. Then next one. Which one of the following is an autosomal dominant disorder? Sickle cell anemia is recessive. Cystic fibrosis, I already told it is autosomal recessive. Tay-Sachs disease is also autosomal recessive. Huntington's disease is autosomal dominant. So the answer here is option 4. Then next one. Which one of the following statement correctly describes type 2p restriction enzymes? They cut nucleotides only from the ends of the DNA. No. They are endonucleases. So they can cut within also. Then they act on only palindromic nucleotide sequence in the DNA. That is correct. They recognize only GC rich sequences, they recognize only AT rich sequences. They both are not correct. So the answer here is option 2, that is B only. Then next is match the items in list 1 with items in list 2. Incomplete dominance. Incomplete dominance is seen in flower color in 4 o'clock plant. So A2. So out of the options given below, 1 and 4 are having A2. The other two options can be eliminated. Then B, recessive epistasis. Recessive epistasis is coat color in mice. B3. So the answer automatically becomes fourth one, option 4. Then dominant epistasis is seen in fruit color in summer squashes, and incomplete dominant and codominance is seen in ABO blood group types. So the answer here is option 4, A2, B3, C4, D1. Then next one. Given below are two statements. In the light of the above statements, choose the correct answer from the options given below. So statement 1. Activation energy is the difference between the energy level of substrate and its transition state during the reaction. That statement is correct. Statement 2. Enzyme increase the activation energy enhancing the rate of the reaction. Actually, enzymes decrease the activation energy enhancing the rate of the reaction. So statement 2 is not correct. So statement 1 is true, but statement 2 is false. So the answer here is option 3. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. In the light of the above statement, choose the most appropriate answer from the options given below. So first one. In assertion, inhibition of succinate dehydrogenase by malonate represents competitive inhibition. That's correct. Malonate and succinate bind at different sites of the enzyme. They have told it is competitive inhibition. So the substrate and inhibitor will bind to the same site of the enzyme, not at different sites. So A is correct, but R is not the correct explanation of A. Okay, third option will be more correct. A is correct, but R is not correct. So answer is option 3. Then next one. Given below are two statements. One is labeled assertion A and the other is labeled as reason R. Assertion A. Bt toxin does not kill Bacillus thuringiensis. Reason. The Bt toxin gets activated due to the alkaline pH of the insect gut. So here both A and R are correct, but R is not the correct explanation of A, because Bt toxin does not kill Bacillus thuringiensis because it is inactive in bacteria. Bt toxin is present in the inactive form in bacteria, so it is not going to kill the bacterium. The Bt toxin gets activated due to the alkaline pH of the insect gut. That is correct. So both A and R are correct, and R is not the correct explanation of A. Then next one. Which of the following techniques can be used to determine the copy number of genes? So chromatin immunoprecipitation, that is used to determine DNA protein interactions. Then northern blotting, it is used to study RNA, and ELISA is used to identify specific proteins. So what is left out? Real-time PCR. So the answer here is real-time PCR. Then next one. Given below are two statements. Choose the most appropriate answer from the options given below. Statement 1. In plants, the genes for smaller subunit of ribulose 1,5-diphosphate carboxylase oxygenase Rubisco enzyme is present in the plastid genome. The genes for larger subunit of Rubisco enzyme is present in the nuclear genome. So here, if you see Rubisco, it is a complex consisting of two subunits. The large subunit encoded by the chloroplast gene and the small subunit encoded by the nuclear gene family. So here they have given the exact opposite of whatever is correct. So both the statements are not correct. So both statements 1 and 2 are incorrect. Answer is option 2. Then next one. Which enzyme catalyzes strand separation at the replication fork? So strand separation is catalyzed by DNA helicase. Answer is option 2. Then next one. Which one of the following small RNAs in plants do not regulate gene expression by cleavage of mRNAs? So it is heterochromatin siRNA. Then next one. Which is a type of multigenic interaction? Lethal alleles, codominance, epistasis, ABO genotype. Epistasis is a type of multigenic interaction. All the others are intraallelic interactions. Then next one. Given below are two statements. Choose the most appropriate answer. Bisexual flowers promote self-pollination. Statement 2. Self-incompatibility and male sterility prevent self-pollination. So flowers containing both the male reproductive parts and the female reproductive parts are called as bisexual flowers. Self-incompatibility is a genetic mechanism that will suppress pollen tube development or pollen tube growth. So both statements 1 and 2 are correct. Answer is option 1. Which is a class of nitrogen containing plant secondary metabolites? Terpenoids, flavonoids, alkaloids, brassinosteroids. So the answer here is option 3, that is alkaloids. Then next one. In a random mating population, the frequency of allele A1 and A2 in the gene pool is 0.8 and 0.2 respectively. In a population of 200 plants, how many plants will have the genotype A1, A2? So they have asked for frequency of the heterozygote. So that is calculated using formula 2 into P into Q. So P and Q they have already given. P is 0.8, Q is 0.2. So 2 into 0.8 into 0.2, that is 0.32. That is in 100. Okay, 32 in 100. So in 200 it will be 64. Answer is option 3. Then next one. During protoplast fusion, the product has the nucleus of one of the parents in which the chromosome elimination took place in subsequent cell division and the cytoplasm was contributed by both the parents. Such a product will be called. So it is called as asymmetric hybrid. So hybrid is whole cell fused with an enucleated cell. That is called as hybrid. And symmetric hybrids, they contain the somatic chromosome of both the parental species. Here they have asked the product has the nucleus of one of the parents in which the chromosome elimination took place in subsequent cell division and the cytoplasm was contributed by both the parents. So that is a case of asymmetric hybrids, because in asymmetric hybrids there are two these are those hybrids which preserve the genetic material of one parent organism. The chromosomal content of the other parent organism or species is lost. That is the case of asymmetric hybrids. So the answer here is option 2, asymmetric hybrids. Then next one. How many clones must you get to make a representative genomic library of a plant with a genome of 1 billion base pairs provided your vector carrying capacity is 10 kb? Remember to allow a redundancy of at least one order of magnitude. So you need 2 million. Answer is option, sorry, not 2 million, option 2 million. Answer is option 2. Then next one. Given below are two statements. Virulence rate of agrobacterian tumefaciens is born on genomic DNA. It is not on genomic DNA, it is on the TI plasmid. The technique used to introduce genes in dicot is TI plasmid infection. That is correct. So first statement is false and second statement is true, and your option is option 4. Statement 1 is false, but statement 2 is true. Then next one. Why is golden rice pale yellow in color? Because it is rich in beta carotene. Golden rice is for vitamin A. So it is rich in beta carotene. Answer is option 2. Then simple sequence repeats are best utilized for. They are best utilized for genotyping. They are favored molecular markers for plant genotyping. Why? Because of their high levels of polymorphism and wide distribution across plant genomes and ease of use. So because of these three reasons, they are usually used in genotyping. Then next one. Given below are two statements. The fragments of DNA generated after cleavage with the restriction enzyme have a phosphate group on their 3' ends and an OH group on their 5' ends. Next statement. Cohesive ends generated by restriction digestion reanneal with complementary DNA fragment. I have explained this in the next slide. So this is the first option what they have told. The fragments of DNA generated after cleavage with their restriction enzyme have phosphate group on their 3' ends and an OH group on their 5' ends. Now if you take eco R1, these are the restriction sequence that it will digest. This is the restriction site where it will digest, and you will get 5' G, then 3' AATTC. So 5' is phosphate, not 3' phosphate group as mentioned in the question. So statement 1 is not correct. If you take blunt end cutter also, here also at the 5' end you have got phosphate group being attached and 3' OH group. So statement 1 is not correct. Now coming to the next statement. Cohesive ends generated by restriction digestion reanneal with complementary DNA fragments. So these are the restriction sites, that is digestion with eco R1. I have taken the same example of eco R1. Once you digest it, you will get two cohesive ends like this for digesting this one, and for after digestion of this one you will get two cohesive ends like this, and they both can form complementary base pairing and reanneal. So option 2 is correct and statement 1 is incorrect. So the answer here will be option 4. Statement 1 is false, but statement 2 is true. Then next one. Given below are two statements. After immobilization, plant cells are subjected to stress to induce secondary metabolite production. That is correct. Then next one, statement 2. Cells derived from different explants in culture always produce the same set of secondary metabolites. That statement is false because different explants will give different sets of secondary metabolites. So cells derived from different explants, that is different explants means one may be stem using a stem as explant, the other may be leaf as explant. The amount of secondary metabolite also will be different from one explant to another. So they don't produce the same set of secondary metabolites. So the answer here is statement 1 is true, but statement 2 is false. Option 3. Then given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Choose the most appropriate answer. So assertion A. A linear DNA molecule containing 3 recognition sites for a restriction enzyme will generate multiple DNA fragments on partial digestion. Partial digestion means you are not digesting it completely. So you can get different fragments after partial digestion. So that is correct. Then next reason. Partial digestion by restriction enzymes lead to heterogeneous population of DNA fragments. Yes, that is also correct. Partial digestion will give you fragments of different lengths. So answer here is both A and R are correct, and R is the correct explanation of A. Then next one. Given below are few statements related to plant tissue culture. Regeneration frequencies of different types of explants derived from the same plant would always be identical. No, but they would be different because chances of somaclonal variation also is there. Different explants, they will have different regeneration frequencies. Then next one. A higher auxin to cytokinin ratio would induce production of roots under culture conditions. That is also correct. Embryos can be obtained from somatic tissues of the plants under in vitro condition. Yes, as in the case of somatic embryogenesis. Then since plants can photosynthesize, addition of a carbon source in tissue culture media is not advisable. Usually glucose or sucrose is added to the medium. So D is incorrect. So the correct option is option 3, B and C only. Then next one. Match items list 1 with items in list 2. Northern hybridization. Northern hybridization is analysis of splice variants. A3. A3 is there only in one option. So option 3 is your answer. Okay, we will see then. Next generation sequencing is transcriptome analysis. Sanger's DNA sequencing is capillary sequencers, and in situ immunolocalization is detection of specific proteins, detection of proteins in specific cell types. So answer here is option 3, A3, B1, C4, D2. Then next one, match items in list 1 with items in list 2. Choose the correct answer from the options given below. Bar. Bar is for is a herbicide resistance. Bar gene, herbicide resistance. A2. A2 is there only in option 4. So 4 is going to be your answer. Meristem culture. Meristem culture is for virus-free plants. Chalcone synthase. Chalcone synthase is related to flower color, and GAI is dwarf wheat. So the answer here is option 4, A2, B1, C4, D3. Then next one. Given below are two statements about hnRNA processing. Statement 1. Capping occurs by addition of methyl adenosine triphosphate to the 5' end. Statement 2. During tailing, about 200 to 300 adenylate residues are added to its 3' end. Choose the correct answer. So capping occurs by addition of methyl guanosine triphosphate at the 5' end. Not methyl adenosine, it is guanosine. So statement 1 is incorrect. During tailing, about 200 to 300 adenylate residues are added to its 3' end. Statement 2 is correct. So the answer here is statement 1 is false, but statement 2 is true. The next one. The vector which is most commonly used in crop improvement is, for crop improvement, it is Agrobacterium tumefaciens or Agrobacterium rhizogenes. So the answer here is option 2. The next one. Lac repressor of E. coli contain dash DNA binding motif. They contain helix turn helix DNA binding motif. Then next one. Knockout mice are usually created by, so chemically mutagenizing a mice and then selecting for mutant offspring. No. Knockout means you disrupt or the function of that particular gene. Knock out. You are throwing it out, just like throwing out or making it non-functional. That is called as knockout. Then next one. Creating a chimera by fusing cells from two different cell lines. That is also not correct. Infecting the mouse with a retrovirus. That is also not correct. So option 4, if you see, transfecting embryonic stem cells with an inactivated gene. That is correct. Then next one. RNAi or post-transcriptional gene silencing is a conserved biological response to the presence of. to the presence of double-stranded RNA? Answer is option 4. Then next one. Which protein is required to seal a nick during Okazaki fragment maturation? It is DNA ligase. Answer is option 1. Then next one. Self-splicing introns are example of. They are ribozymes. Answer is option 2. So which of the following can be a restriction site for type 2 restriction enzyme? So it should be palindromic sequence. Palindromic means sequence in one strand is identical to the sequence in the complementary strand but in the same direction. So if the sequence is AAGG, if you see the first option in 5' 3' direction, it should read the same AAG in the 5' 3' direction in the complementary strand or the second strand. But first option it is not like that. Then next one. AGTC 3' to 5'. So 3' to 5' should be AGTC, but here it is not so. So option 2 is also not correct. If you see options C3, GGCC, GGCC, option 3 is correct because it is reading the same in both the strands, and in the complementary strand it is reading in the same direction itself, that is 5' to 3'. So in the first strand 5' is GGCC to 3', and in the second complementary strand it is GGCC 5' to 3'. So it is a palindromic sequence, so it can be cut by a restriction enzyme type 2. Then next one. A mutant bacterial cell has a defective amino acyl synthetase that attaches a lysine to tRNAs with anticodon AAA instead of the normal phenylalanine. So tRNAs with anticodon AAA, they usually code for phenylalanine, but since the bacteria is having defective amino acyl synthetase, it attaches lysine to these tRNAs instead of phenylalanine. So during protein synthesis, what will happen? None of the proteins in the cell will contain phenylalanine. Nothing like that. There will be phenylalanine because there are other codons which are coding for phenylalanine. Not only AAA but other codons are also there which code for phenylalanine. Lysine will partially replace phenylalanine at certain positions. That is correct. So option 2 is correct. Then next one. A double-stranded genomic DNA has 40% A plus T content. What is the average size base pairs of the fragments generated by digestion with the restriction enzyme He3-GGCC? The answer here is 1, 2, 3, that is option 2, and I have done the explanation in the next slide. So they have given A plus T as 40%, therefore G plus C will be 60%. So probability of finding A is 0.2, probability of finding T is 0.2, and probability of G is 0.3 and C is also 0.3. So for the sequence GGCC, probability of G at position 1 will be 0.3, G at position 2 will be 0.3, C at position 3 will be 0.3 and C at position 4 will be 0.3. So multiply all the probabilities, you will get 0.0081, and average number of fragments will be 1 divided by reciprocal of this particular answer, that is 0.0081. You will get it approximately as 123.45 or approximately 123. So the answer is option 2. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. In the light of the above statement, choose the correct answer from the options given below. So assertion A. Attenuation of the tryptophan operon is possible in bacteria. That is correct. Transcription and translation are coupled. That is also correct. And attenuation of tryptophan operon is possible in bacteria because transcription and translation are coupled. So both A and R are true, and R is the correct explanation of A. Answer is option 1. Then which is the correct order of progressively higher level of chromatin organization? So you have got nucleosome, 30 nanometer chromatin fiber, looped domain. So the answer also is the same thing, that is A, B and C. Nucleosome, 30 nanometer chromatin fiber, looped domain. So the answer is option 1. Then next one. The following diagram represents a restriction enzyme site. This is the site. After digestion, cohesive ends will be formed. Of course, you will get cohesive ends over here. If you cut over here, it will cut over here. You will get cohesive ends. These cohesive ends will have phosphate group at 3' end and 5' ends. Again, the statement is incorrect. Just previously, I told that the cohesive ends or after digestion with restriction enzyme, you will have phosphate groups at the 5' end and 3' end will have OH groups. So the statement 1 is correct, but statement 2 is incorrect. So the answer here is option 3. Then next one. The following figure shows agarose gel electrophoresis of linear DNA after digestion with BAM-H1 in lane C and ECO-R1 in lane D. The uncut DNA is shown in lane B. So the total size of the DNA is 1500 base pairs. So these are the BAM-H1 digestion products. These are the ECO-R1 digestion products. Statement 1. This DNA has 2 restriction sites for BAM-H1 and 1 restriction site for ECO-R1. That is correct. If you see the figure over here, you can see that BAM-H1 cuts at 2 different positions and ECO-R1 cut only at 1 single position. Double digestion with BAM-H1 and ECO-R1 yield 5 fragments. Need not be, you can get 4 also. So in the light of the above statement, choose the correct option. So both the statements 1 and 2 are true. No. Statement 1 is true, but statement 2 is not. So the answer here is option 3. Then next one. During replication, DNA polymerases distinguish between ribonucleoside and deoxyribonucleoside triphosphates while synthesizing new DNA strand. Although RNTPs are present at approximately 10-fold higher concentration in the cell, they are incorporated at a rate that is more than 1000-fold lower than DNTPs. This is because during replication, DNA polymerases, they can distinguish between RNTPs and DNTPs. And RNTPs, they are usually present at higher concentration than the DNTPs in the cells. So how does this DNA polymerase distinguish between these RNTPs and DNTPs? That is because of steric exclusion of RNTPs from the DNA polymerase active site. Answer is option 3. Then next one. Regulation of lac operon is negative because regulation is negative because expression of the operon is blocked by an active repressor. Answer is option 3. Next one. Recombinants with insertional inactivation of beta-galactosidase are identified by their ability to. So upon cloning, beta-galactosidate will not be expressed and you will not get blue color colonies in the presence of XGAL. And XGAL, it is a chromogenic substrate. So recombinants with insertional inactivation of beta-galactosidase are identified by their ability to produce color in the presence of a chromogenic substrate. Answer is option 4. Then next one. The level of expression of recombinant proteins can be gauged by. So they can be gauged by Western blotting. Answer is option 4. Then next one. The percentage of sequence identity for the following aligned sequences. So they have given a sequence there and you are asked to calculate the sequence identity. So how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 protein sequences, 10 sequences are there. And out of that, how many are matching exactly? 1, 2, 3, 4, 5, 6 and 7. 7 are matching. So 7 out of 10 by 100. That is 70%. Answer is option 4. Then next one. At the time of completion of protein synthesis, the sequence in which they leave the ribosome is. So it is polypeptide chain, then tRNA and then finally mRNA. Answer is option 3. Then next one. Subtractive hybridization is useful too. So it is useful to identify set of genes that are only expressed under certain conditions. Answer is option 4. Then next one. NK cells are immunologically significant as they recognize and kill. So, as they recognize and kill cells with down-regulated MHC1 molecules. Answer is option 3. Then the primary function of class 1, class 2 MHC molecule is. So it is binding peptide antigens for recognition by antigen-specific receptors on T cells. Answer is option 1. Then next one. What is the advantage of using the neo gene to disrupt the function of a gene in knockout mice? The neo gene, it provides a selectable marker. Answer is option 3. Then next one. The selection of cells whose T cell receptors respond to self-MHC is known as. It is called as MHC restriction. Answer is option 2. All these are just direct questions. That's why I am not explaining much.